All right, Mr. Herbert, are you ready for your medicine? Get it away from me. You know I don't like the lemon flavor. Well, the lemon flavoring was all we had, so you're gonna have to suck it up this time. This is this is humiliating. I used to be someone. Before I was ridden to this god-forsaken wheelchair, I was at the top of my field in forensic science. Worked on so many cases, I can't even remember. Putting away every wannabe John Schneeberger and Donald Klein there was. Wow, that sounds interesting. Is there any case in particular you do remember? Only one. Spring, 1998. I was a lead forensic scientist at the University of Ottawa, where my team and I were faced with a task brought to us by the police department. Over the past few months, kids ages 2 to 13 living in Toronto were all falling ill, each of them experiencing a similar set of symptoms, followed by stroke-like episodes. Our team took blood samples from all over, the results somewhat inconclusive. You see, most of the patients had notably high blood pressure, but not enough for it to be hemorrhagic. Nor could we attribute the strokes to be ischemic. They all seemed healthy, but not enough cholesterol was in any of their bodies to cause blockage. Yet despite all these factors, they all seemed to suffer from these apparent strokes. We noticed from most of the police reports, however, that these children had felt an overwhelming sense of tiredness via lack of breath. So we thought it was metabolic. We decided to bring some of the kids in for tests, and what we found was shocking. MR spectroscopy revealed red jagged lines of lactic acid built up in muscle tissue, brain tissue, kidneys, eyes, you name it. So many organs affected, and for a long time we thought it was a cancer. It wasn't, but it did explain a lot of the symptoms they were all experiencing. But what are the strokes? Not a lot died from this mysterious illness, but there were a few dead kids. And although that was very saddening for us, we were able to make best of the situation and use some tissue to analyze potential threats, potential causes. And what we found in the brain tissue is a deficiency in nitric oxide, which was an important retrograde neurotransmitter responsible for moderating blood flow throughout the neocortex. So, with lactic acidosis in mind, along with this deficiency in nitric oxide, we were able to chalk this up to potentially some kind of cause in the failure of the oxygenation of tissue, poor oxygenation of tissue, which led us to metabolism, which terrified us. Furthermore, all the organs that were affected by this lactic acid buildup were the most high-functioning metabolic components of the body. So if anywhere, this is where we're going to find the problem. Taking a look at the blood samples again, we noticed completely normal oxygen levels, but an extremely high NADH concentration. Most cases with conditions like these means some kind of complication may have arose in the mitochondria, compromising a certain pathway. However, it was this lactic acidosis that made it seem all off. It could have been some form of mutation in the large enzyme complex making up the ETC, which would explain the poor oxygenation of the tissue, fatigue from impaired oxidative phosphorylation, and the NADH buildup inhibiting potential parts of the mitochondria. But you have to understand that the high energy electrons would have caused a buildup within complex 4, having it be unable to receive oxygen as a final electron acceptor, and so on the process goes. But that didn't completely explain the lactic acid buildup. You see, when ETC is compromised, and we saw that, aerobic respiration begins to break down in a way. ETC is not happening, nor is oxidative phosphorylation in the mitochondria, but at the same time, other processes would halt as well. Except for anaerobic respiration, cells in the muscles would use pyruvate from glycosis and redirect it into a new metabolic pathway, lactic fermentation. Lactate would form in the muscles and eventually be converted to pyruvate via an enzyme, pyruvate dehydrogenase. But it wasn't. We saw that with the red jagged lines from the MR spectroscopy, but we didn't know. Pyruvate also wasn't going into the Krebs cycle at all, likely due to the inhibition of isocitrate dehydrogenase by the surplus of NADH concentration in the blood. So these proteins involved in the removing waste of vital tissues following anaerobic respiration, which was induced by this compromised ETC, could have explained maybe some kind of mutation in protein makeup. It was more than just the complex 4, it was proteins on a larger scale. So we were thinking to ourselves, Could this be something with DNA, mitochondrial DNA, the buildup of proteins via transfer RNA? That was likely at play. The proteins that make up ETC and lactic removal enzymes were clearly beginning to take form of its dysfunction in these young kids, and we were able to conclude, following further testing on a genetic level, that this was in fact MELAS, associated with the mutation 3423AG of the mitochondrial DNA gene. We asked mothers about potentially any family history with mitochondrial encephalopathy, lactic acidosis, stroke-like episodes, but it turned out to be nothing. I mean, we even took genetic samples from the parents, relatives, grandparents. Nothing came up. But how? How could there be so clearly a mitochondrial DNA mutation in these children, yet none in their family histories? I mean, you could argue that there was some kind of grand coincidence that the cells happened to mutate based on error alone, which led to a domino effect of awfulness, but it was MELAS. We saw it was MELAS in all of these kids. The MTTL1 gene was clearly affected, which was responsible for transfer RNA. So that explained the myriad of the protein dysfunctions in the body. But then it hit me. If none of the families are affected, but all of these kids are affected, each of them experiencing a similar set of symptoms with organs also similarly compromised, it thought, well, what if they're somehow all related? And if they're all related, that may have meant that they all have the same mother, which would explain the mitochondrial DNA inheritance. I was determined at this point, and I mean, the loss of so many kids from this mysterious illness, it choked me ruthlessly with guilt. The better part of this investigation forcing me to turn to alcoholism, but that was beside the point. I ran some independent tests in the lab to see if the children were actually related to their parents. Turned out none of them. I asked if they were orphans, if they were adopted. No, parents were shocked. They didn't know about this. None of them were related to their quote-unquote biological parents. Furthermore, I was able to conclude that each of the affected children, there were 11 at the time, excluding the 8 dead, they were all biological siblings. I took this to the police, and in about 5 months, they were able to find the biological mother. Her name? Donna Hannings. Apparently, during the 1980s, she worked as a nurse who overlooked the incubation of newborns, having been employed by over 11 to 12 hospitals over the country. According to a confession made by her after detainment, it was discovered that she had been replacing healthy children with her own babies, completely unaware of the mitochondrial DNA she would inherit to these children. What she did with the real children, well, we didn't know, as before we were able to question her further, she collapsed to the ground, dying via cyanide poisoning in the hospital about an hour later. Investigators found cyanide capsules hidden beneath her mattress in her apartment, which would potentially explain this deliberate poisoning, suicide, if you will. Why she did this? Donna Hannings? I don't know. A lot of people chalk it up to mental illness, but I really couldn't care at that point. I was so entangled with the mystery of it all. It swallowed me whole, really. I was devastated and brought into a drunken mess that you see before you. It wasn't long until I quit, and many years have gone by, and decades even. I still remember it, all of it, haunting me. As for those 11 children, they're dead. Lives dwindling off in the past couple of years. MELAS is often fatal, with people dying on average 11 years after experiencing initial symptoms. Wow. I can't begin to feel your pain. It's all right. Thank you. I appreciate you listening to me, Mrs. Hannings. Jane Hannings. Well, let me go get your medicine. Hannings? Oh. Ooh.